Section: Introduction. In this section, we introduce how the human brain processes sensory information, creating a complex understanding of our environment. While we have made great strides in understanding visual, linguistic, and auditory perception, the area of olfactory perception remains less explored. There is no clear framework for how we perceive odors, making it a challenging topic, especially since there are no widely accepted methods to describe odors effectively. We note that few studies have linked chemical structures to olfactory perception, and the use of deep neural networks for processing olfactory stimuli is still in its infancy. Training existing models often requires significant effort from experts to label data. However, transformer-based models have emerged as a significant advancement in machine learning, reducing the need for extensive labeling by using implicit supervision. These models have shown strong performance in various fields, including image and language processing, and have recently begun to show promise in encoding chemical structures. We pose the question of whether the representations of odorant chemical structures from transformers trained on chemical data align with human olfactory perception. To explore this, we utilize Molformer, a cutting-edge transformer model pre-trained on chemical structures. Our findings indicate that the representations extracted from this model can predict expert-assigned labels for odorants, forecast continuous ratings provided by human participants, and show a strong correlation with both human perceptual similarity ratings and relevant physiochemical descriptors. Remarkably, these results are achieved even though the models were not specifically trained to predict human olfactory experiences. This represents the first empirical study assessing the alignment between transformer-encoded odorant representations and human olfactory perception. In the related work section, we highlight the growing interest in predicting olfactory perception from molecular structures, primarily driven by the neuroscience community. Previous efforts have utilized standard chemoinformatic representations to model olfactory perception. For instance, Snitz et al. Developed a framework based on molecular structural features to predict perceptual similarities between odor pairs, identifying key features among numerous physiochemical descriptors. Ravia et al. Later expanded this model to include perceived intensity, reporting improved correlation with their weighting approach. We also discuss recent advancements in deep neural networks for odorants, such as the Principal Odor Map (POM) model proposed by Lee et al., which uses a message-passing graph neural network. This model, trained on a dataset of about 5,000 molecules with expert-labeled descriptors, outperformed previous models but still relied on subjective expert evaluations, which can introduce biases. Lastly, we mention the emergence of large-scale pre-trained models, like Molformer, which can perform various tasks using vast amounts of unlabeled data. Molformer employs a transformer architecture and is trained through a self-supervised approach on multiple datasets, focusing on masked token prediction. In the method section, we will detail the datasets we used, our approach to extracting both machine and human representations, the main model and baseline methods, and the evaluation metrics for assessing performance. Our experiments are designed to be resource efficient, primarily using linear models that do not require extensive computational power. Section Summary. In this section, we explore the alignment between odorant representations from the Molformer transformer model, pre-trained on chemical structures, and human olfactory perception. Our findings indicate that these representations can effectively predict expert-assigned labels and human perceptual ratings, marking the first empirical study on this alignment in olfactory perception. Section: Datasets. In this section, we describe the datasets we utilized, all of which are publicly available from the Perfume repository. First, we used the Leffingwell GoodScent dataset, which is a combined version of the GoodSense and Leffingwell datasets. This dataset includes 4,983 molecules, each associated with 138 expert-labeled descriptors, such as creamy or grassy. Each odorant can be linked to multiple descriptors. Next, we examined the Sagar dataset, which features ratings of 160 odorants by 3 human participants across 15 common descriptors, plus 3 additional descriptors that varied among participants. We focused only on the common descriptors and used the normalized ratings, which range from -1 to 1, averaging them for our analysis. We also analyzed the Keller dataset, which contains ratings for 480 diverse molecules from 55 participants, evaluating 23 descriptors. Participants rated the odorants using a slider, and we averaged these ratings across all participants for further analysis. The Rabia dataset includes similarity ratings for 195 unique pairs of MC odorants and monomolecules from 94 participants. We averaged the similarity ratings while ignoring the intensity of the odorants. Additionally, we used the SNITS dataset, which features similarity ratings from 139 participants for 359 unique pairs of odorants. Participants rated the similarity of 2 distinct odorants, and we averaged these ratings across all participants. For odorant representations, we defined odorants as either single molecules or mixtures, referred to as MC odorants. We extracted representations using our main pre-trained model, olfactory, which encodes SMILES strings into 768-dimensional vectors. For MC odorants, we averaged the representations of the individual molecules. We also employed OpenPalm, a supervised learning model based on a graph neural network, trained on the GSLF datasets to predict odorant perceptual labels. We used a publicly available version of this model, training it for 150 epochs and extracting representations from its penultimate layer, resulting in 256-dimensional vectors for each dataset. The distance angle model, DAM, was another tool we used, which predicts similarities between pairs of odorants based on 21 physiochemical descriptors. We extracted these descriptors, discarding 6 due to missing values, and used the remaining 15 to measure similarity or to create a linear mapping to the perceptual representation space. Finally, we gathered perceptual representations from human participants exposed to odorant stimuli. Experts labeled the odorants, linking them to multiple labels, while non-expert participants provided ratings based on predefined descriptors. We averaged these ratings to form matrices representing the perceptual data. Participants also evaluated the perceived similarity between pairs of odorants, leading to averaged vectors for these comparisons. Section Summary. In this section, we describe the datasets used for our analysis, including the Leffingwell-Goodcent dataset with 4,983 molecules and expert-labeled descriptors, the Sagar dataset with ratings from human participants, and others that provide similarity ratings among odorants. We also outline our methods for extracting odorant representations using models like molFormer, OpenPalm, and DAM, which utilize various molecular and perceptual data to create structured representations for further analysis. Section: Alignment between perceptual and odorants representations. In this section, we measure how similar different representation spaces are. When possible, we directly compare them. Otherwise, we train a linear model to predict perceptual representations for each odorant. We use a method called nested 5-fold cross-validation to fine-tune the models and evaluate their performance on a test set that we set aside during training, using an 80% to 20% split. This process is repeated 30 times with different splits. We introduce our main evaluation metrics here. The micro-averaged ROCAUC score helps us assess how well each model performs in multi-label classification by combining true positives, false positives, true negatives, and false negatives across all classes. The normalized root mean squared error, NRMSE, measures the difference between actual and predicted values in regression tasks, normalized by the range of true observations. The Pearson correlation coefficient indicates the linear relationship between predicted and actual values, calculated as the ratio of covariance to the product of standard deviations. Next, we evaluate whether representations from pre-trained chemical models can predict human olfactory experiences, even though they weren't specifically trained for this. We first focus on predicting expert-assigned labels for odorants using linear mapping from representations to perceptual descriptors. Then, we predict continuous scores given by human participants and direct similarity scores from odorant representations. To evaluate Mulformer's ability to predict expert-assigned labels, we map its representations to those from the GSLF dataset. We reduce the dimensionality of these representations to 20 using PCA and standardize each feature. We train separate logistic regression models for each descriptor, repeating this 30 times to assess uncertainty. For the DAM model, we skip dimensionality reduction, and for OpenPalm, we directly use its predictions without retraining. Our results show that Molformer achieves high ROC-AUC scores in odorant classification, outperforming DAM but falling short of OpenPalm, which is trained with supervision. We also compare the perceptual details captured by Molformer and OpenPalm. The first two principal components reveal that both models show similar representations, with Molformer capturing perceptual relationships without using perceptual labels during training. For predicting continuous ratings based on predefined descriptors from human participants, we train separate linear regression models with lasso regularization for each descriptor, again reducing dimensionality to 20 using PCA. We repeat this with 30 different train test splits. Our results indicate that none of the models show high correlation overall, with Molformer slightly underperforming compared to OpenPalm. However, it performs better than DAM for one dataset but worse for another, where DAM outperforms OpenPalm. Overall, Molformer performs comparably to the supervised models in predicting ratings for each descriptor, demonstrating a similar level of alignment despite lacking supervision in its training. Section Summary. In this section, we evaluate the alignment between perceptual and odorant representations using various models, including Molformer, by measuring performance through metrics like micro-averaged ROC-AUC, NRMSE, and Pearson correlation coefficient. Our results indicate that while Molformer shows promising alignment with human olfactory experiences, it generally underperforms compared to the supervised OpenPalm model, yet still captures perceptual relationships without explicit training on perceptual labels. Section: Representational Similarity Analysis. In this section, we conduct representational similarity analysis to assess how well the odor similarities captured by Molformer align with those reported by human participants. We encode each odorant using Molformer and baseline models, then calculate the cosine similarity between the representations. We also compute the Pearson correlation between the similarity scores from our models and those from human participants in the RAVIA and SNITS datasets. Our findings, illustrated in a figure, indicate that Molformer effectively captures information relevant to human olfactory perception, even without direct training on this data. We observe a strong correlation between the perceptual and odorant representations for both datasets, with values of approximately 0.64 and 0.66, both statistically significant. When compared to baseline models, Molformer performs similarly to the OpenPalm model and significantly better than the DAM model, suggesting that these models may struggle to extract odorant similarities effectively. Furthermore, Molformer excels at identifying similarities between odorants rather than mapping them to predefined descriptors, likely due to its ability to reflect human-like perceptions without the biases of subjective language. Next, we explore whether the layer depth in Molformer affects representational alignment. We repeat our analysis for each layer and find that deeper layers show better alignment with high-level perceptual representations. We also evaluate whether Molformer extracts relevant physiochemical features from chemical structures. To do this, we train 15 linear regression models to predict individual physiochemical descriptors from Molformer's representations and assess the correlation between predicted and actual values. Our results show that Molformer accurately predicts 13 out of 15 descriptors, performing as well as or better than the OpenPalm model. We analyze how this alignment varies across layers, finding that while alignment with chemical features decreases with depth, alignment with perceptual features improves. This pattern aligns with established principles in vision models, where lower layers capture basic features and deeper layers represent more abstract concepts. However, further research is needed to fully understand this hierarchical structure. In our discussion, we highlight that our study reveals a close alignment between odorant representations from MOL former and human olfactory perception, suggesting that chemical structures can predict odorant perception effectively. We also show that our model can predict relevant physiochemical descriptors, providing valuable insights for future research in chemistry and neuroscience. To evaluate alignment from different angles, we conduct 3 experiments. First, we use a dataset with expert labels to assess the model's ability to predict binary labels for odorants, where MOL former performs well. Second, we analyze datasets with average ratings from human participants, which present more variability. Here, MOL former performs comparably to supervised models. Lastly, we examine direct similarity scores between odorants, finding that MOL former aligns well with human-provided scores, suggesting that predefined descriptors may not accurately reflect true odorant similarities. We also analyze the alignment of MOL former representations with physiochemical descriptors, noting that while most features align well, some do not. Our findings indicate that as we progress through the model layers, alignment with physiochemical descriptors decreases, while alignment with perceptual features increases, consistent with principles observed in vision models. We acknowledge the limitations of our work, particularly the lack of consideration for the intensity or concentration of individual molecules in odor mixtures, which could enhance alignment in future studies. Additionally, our research is limited by the datasets available, which often lack sufficient variation for continuous rating tasks. We also focused on average rating scores rather than individual subject ratings. Looking ahead, we plan to use our findings to develop better models of olfactory perception, focusing on unsupervised models trained on chemical structures to identify key features for predicting perception without human biases. We also aim to investigate the mechanisms behind olfactory perceptions derived from chemical features, leveraging the alignment trends across model layers for insights. Finally, we intend to evaluate the alignment between representations from chemical structure-trained transformers and fMRI data to deepen our understanding of olfactory perception mechanisms. Section Summary. In this section, we evaluate the alignment between odorant representations encoded by MOL former and human olfactory perception, finding a strong correlation with human similarity ratings and demonstrating that MOL former outperforms baseline models in capturing perceptual similarities. We also explore the model's ability to predict physiochemical descriptors, revealing that while alignment with these features decreases in deeper layers, alignment with perceptual representations improves, suggesting a hierarchical structure similar to that observed in vision models. Section: Acknowledgement. In this section, we express our gratitude to those who contributed to our work. We thank our colleagues for their support and valuable insights. We also appreciate the funding agencies that made this research possible. Lastly, we acknowledge the contributions of our families and friends for their encouragement throughout this process.